Make an appointment with your subject, preferably between 9 and 11 a.m., and ask the subject to refrain from eating 30 minutes prior to saliva collection. Have the subject rinse their mouth with water and discard. Five minutes after rinsing, ask the subject to spit into a pre-chilled 50 milliliter Falcon tube. Please note that Falcon tubes should be kept on ice for at least 10 minutes before collection. Remind the subject to refrain from coughing up mucus and no nasal mucus, only saliva from the mouth. Collect 5 milliliters of saliva. Do not take more than 30 minutes to collect the saliva and keep the collected saliva on ice. Centrifuge the collected sample at 2600 G for 15 minutes at 4 degrees centigrade. Incomplete separation may occur. If so, centrifuge for an additional 20 minutes. During centrifugation, label the cryotube with the appropriate ID code, following this example. Label SUP or PROT and the vial number. SUP indicates RNA, and PROT stands for protein. Date the sample. Using a 1-to-1 ratio, add appropriate reagents to 2 sets of sample vials. Sample 1 for RNA analysis and Sample 2 for protein analysis. The Sample 1 RNA analysis should be given a white cap. Add 1 microliter superase in. Keep the superase in reagent on ice during use. Superase in needs to be stored at -20 degrees centigrade. Then, pipette saliva supernatant into the cryotube. The Sample 2 protein analysis should be given a yellow cap. Add the following reagents. 1 microliter aprotinin. Obtain stock solution kept at 4 degrees centigrade. Keep on ice during use. 3 microliters Na3OV4. Obtain aliquots from -20 degrees centigrade. Thaw on ice and keep on ice during use. Leftover reagent may be stored at 4 degrees centigrade and then discarded after 1 week. 10 microliters PMSF. Obtain aliquots from -20 degrees centigrade. Keep on ice during use, then discard after 4 weeks. Then pipette saliva supernatant into the cryotube. Save the remaining pellet into a cryotube with a blue cap. Transport samples for storage to the -80 degrees centigrade refrigerator on ice. Scientific teamwork depends on consistent methods, and confidence that saliva testing results are consistent and accurate is essential for research and for the long-term development of successful biomarkers.